Hello everyone. Let's get started. Today we're going to be continuing our conversation of microbial genetics. We'll be talking about the processes of transcription and translation. If you remember from last time, we said that DNA replication is a process that makes new DNA from existing DNA. And it's a process that uses a semi-conservative mechanism. That means that each of the old strands of DNA are used to make new DNA, so that once DNA replication is complete, each of the two molecules of DNA contain one old strand and one new strand. We said that we are able to perform DNA replication now in vitro using a technique called polymerase chain reaction, or PCR. We also said that DNA replication is not the only thing that a living microbial cell does with its DNA. Remember, microbial cells are also using their DNA to perform what we would call gene expression. And all gene expression is a combination of the two processes that are mentioned in the central dogma of biology: the process of transcription and the process of translation. The central dogma tells us that DNA is used to make RNA through this process called transcription. And then RNA is used to make protein through the process of translation. Proteins are then what give organisms their traits. Now, when we say that DNA is used to make RNA, we're not being very specific there, because all RNA is made through this process called transcription. And today we'll talk about the forms of RNA, including mRNA, tRNA, and rRNA. All three of them are produced, as well as a couple of other forms of RNA that we won't have an opportunity to talk about in this course. All of them are produced through the process of transcription. It's one of those types of RNA, the mRNA, that is used to make protein through the process of translation. So we're going to get a little more specific today about the central dogma and about these two critical processes that make up what we call gene expression. So, DNA is used to make RNA, and there are three primary types of RNA in living cells: mRNA, rRNA, and tRNA. mRNA is messenger RNA, and messenger RNA serves as the transcript of a gene in DNA. So think about mRNA as being the transcript that is produced in a courtroom, for example. As all of the proceedings go on in the courtroom, and of course those proceedings are done verbally, there's a person in the courtroom, the court reporter, who is making a transcript, who is recording all of that important information into a written document that we would call the court transcript. You can make an analogy in the cell, because there's lots and lots of things that are happening in a DNA molecule. There are lots of genes in a DNA molecule that have lots and lots of information in them. What mRNA is a transcript or a recording of one of those genes. rRNA, or ribosomal RNA, is the type of RNA that makes up part of the ribosome. rRNA is required in order for the ribosome to function correctly. And remember that ribosomes are the machinery that help perform the process of translation. Finally, tRNA is transfer RNA, and transfer RNA is a middleman of sorts in the process of translation. It's tRNA that transfers or delivers, if you will, the amino acid to the ribosome according to each one of the codons that is present in the mRNA molecule. Remember, in DNA, there are groups of bases that make up the protein coding region of a gene. And the mRNA that gets made from that gene is also going to have bases in it that contain the code to build protein. tRNA is going to be the molecule that delivers the correct amino acid for each group of 3 bases, or each codon that's present in that messenger RNA molecule. Let's take a minute and talk about RNA as a molecule, especially in comparison with the DNA molecule. RNA, like DNA, is an abbreviation. RNA stands for ribonucleic acid. Just like DNA, RNA is essentially a polymer. It's composed of many individual subunits that are bound to each other to make a nice long strand. The subunit of RNA is also a nucleotide. It's just a different kind of nucleotide than what you find in DNA. It has a lot in common with the DNA nucleotide, however. It still has a sugar molecule in it, it has a phosphate group in it, and it has a base. It's just that those parts of the ribonucleotide have a couple of structural differences. Let's take a look at those. The sugar that's found in a nucleotide in DNA is called deoxyribose, while the sugar that's found in a nucleotide in RNA is simply ribose. We'll take a look at those two sugars side by side on the next slide. In a nucleotide in RNA, the base that can be attached is either an A, or a U, or a G, or a C. In the nucleotide in DNA, there is no U, there's no uracil. Instead, there's T, or thymine. So 3 of the 4 bases are the same in DNA and RNA. It's just the 4th base that's different. The 4th base is thymine in a DNA nucleotide, and uracil in the RNA. The last structural difference is the fact that while DNA is a double-stranded molecule in a living cell, RNA exists as a single-stranded molecule. It's not that RNA stays as a linear structure, it doesn't. What happens is the single strand of RNA that gets produced will fold back on itself. It will hydrogen bond with itself from A to U and G to C. And we'll take a look at that in just a minute. Over here on the right-hand side, you can see a drawing of the nucleotide that is found in RNA. This represents the sugar, again, ribose. This represents the phosphate group, and this represents the nitrogenous base, or the nitrogen-containing base. This base might be a guanine, or a cytosine, or an adenine, or a uracil. On this slide, we can see these structural differences that we just talked about. Up at the top, you're looking at a side-by-side comparison of the chemical structure of the sugar found in these two forms of nucleic acid. This is deoxyribose on the left, and this is ribose on the right. Both of these sugars have a ring structure to them. Both of these sugars have the same numbering of the carbons, from the first carbon, the 1 prime, to the 2 prime, 3 prime, 4 prime, and 5 prime, which is the one that's off of the ring. The difference between the sugars is on the 2 prime carbon. In DNA, there's just a single hydrogen group attached to that 2 prime carbon. In ribose, in RNA, there's a hydroxyl group, OH. So if you compare this OH to this H, essentially what's happened is an oxygen has been removed. So we refer to this deoxygenated form as deoxyribose. Down at the bottom on the left, you can see a side-by-side comparison between thymine and uracil. The only difference between these two bases is right here. In thymine, there's what we call a methyl group attached to one of the carbons in the central ring. That's a CH3. In uracil, that methyl group is gone, and all you have is a single hydrogen. That's it. That's the only difference between those two bases. And down here on the lower right, you can see the difference in a single-stranded and a double-stranded molecule. The difference between the double-stranded DNA molecule, again what we refer to as a double helix, and the single-stranded RNA molecule. In both of these molecules, we talk about the sugar phosphate backbone and the bases that are sort of jutting off of it. In DNA, because there are two strands, you have two sugar phosphate backbones, and the bases are oriented off the both of those strands so that they encounter each other, they sort of bump into each other, and that's what allows them to hydrogen bond with each other, A with T and G with C. RNA is a single-stranded molecule. So what will happen with the RNA, where possible, is it will fold over and it will hydrogen bond with itself. Again, where it can, where bases can match up with each other in a complementary manner, A with U and G with C. The RNA molecule is made during this process called transcription. All types of RNA are made during transcription, but since we're talking about gene expression today, we'll focus on messenger RNA. There are actually 3 steps to the transcription process, and I've got them listed in uppercase letters throughout this slide. There's a phase called the initiation, then there's an elongation, and then there's a termination. We'll talk about each one of these separately. Transcription, the process of building messenger RNA on a DNA molecule, requires an enzyme, as you probably guessed. Remember, when we build new DNA from old DNA in a cell, we use an enzyme called DNA polymerase. When you build RNA from DNA in a cell, we use an enzyme called RNA polymerase. One of the biggest differences between DNA replication and transcription is that in DNA replication, remember, the microbial cell is copying the entire DNA molecule. But during transcription, it's only one gene that is being used to make RNA. And recall that a gene is a length of the DNA molecule that has two very different kinds of code in it. Some of the code, some of the letters, the bases, are a code to build a protein. But the rest of the letters, the rest of the bases, are regulatory code. They serve to tell when the protein is made, how much of the protein is made, and so on. So the regulatory region of a gene becomes very important during transcription. And what will happen is this RNA polymerase enzyme is going to come in and interact with the gene first in that regulatory region. There's a very particular location in the regulatory region called a promoter. And it's the promoter that serves almost like a landing strip for this great big enzyme called RNA polymerase. The RNA polymerase enzyme is going to land on the promoter and bind there. And when it does that, it will be set up in space so that as it begins the process of transcription, it will start with the very first base of the protein code. This, again, this interaction with the promoter is initiation. Just like we saw with the DNA polymerase enzyme, the RNA polymerase enzyme is going to travel down this strand of DNA and it's going to build an RNA. It's going to add ribonucleotides one at a time in a complementary way. Where there's an A in the DNA, the RNA polymerase enzyme is going to place a U. Where there's a T in the DNA, the RNA polymerase will place an A. Where there's a G, the enzyme will place C. And where there's a C, the enzyme will place G. As it travels down that DNA strand and builds this RNA molecule, the RNA polymerase is only working in one direction, just like the DNA polymerase enzyme. You can only build RNA from 5' to 3'. Remember what that means when we say this is the RNA polymerase enzyme can only add new nucleotides onto the 3' hydroxy group of the sugar of the last nucleotide. It has to have that hydroxy group, that OH group, jutting off as a site to build the bond between the existing RNA, what's been made so far, and the new ribonucleotide that it's bringing in. So now we know that all nucleic acid can only be made in one direction, and that direction is 5' to 3'. Now, unlike DNA replication, transcription proceeds in only one direction. Remember, during DNA replication, when the replication bubble opens, the forks on either side of that replication bubble are going to continue to open as the new DNA is made. So DNA replication occurs in two directions. Our RNA production through transcription is different. It's only going to happen in one direction along the length of the protein coding region of the gene. And then it's going to end. And that ending process is called termination. Transcription is going to stop when the RNA polymerase enzyme reaches a part of the DNA called the termination signal. We'll talk about termination in just a minute. On the next couple of slides, we're going to look at some drawings of the transcription process. What you're looking at the top of this slide is a double-stranded molecule of DNA. Now, this double-stranded piece of DNA does not represent the entire DNA molecule inside a cell. Instead, it represents just a piece of that DNA that happens to be a gene. Let's take a look at this DNA drawing. We can see these two turquoise strands, this one down at the bottom and this one up at the top. Notice that one end of the top strand is labeled 5'. The other end of the top strand is labeled 3'. That's telling us how that strand is oriented. On this end of the strand, the 3' carbon is exposed with its hydroxy group, and on the other end, the 5' carbon is exposed, attached to the phosphate group. The second strand of DNA is anti-parallel to the top strand. The 3' end is over here, and the 5' end is over here. In between the two strands, we can see these little lines coming off perpendicular to each of the strands. Those represent the bases. You can't see which one is an A and which one is a T or a G or a C, but we know that from strand to strand, the pair of bases, or the base pair, is either A and T or G and C. Now I want you to notice how this end of the double-stranded DNA molecule is colored yellow and is labeled transcribed region. Over on the other end, we can see some areas that are white and some areas that are green. This is the regulatory region of this particular gene. And they're showing us that from right about here to all the way over here, this is the promoter. This is the part of the regulatory region where the RNA polymerase enzyme first comes in and sort of lands like a ship, maybe a spaceship docking. There are two areas in the promoter, the areas that are highlighted in green, that represent exactly where that enzyme is going to come in and bind. There are two different recognition sites, two places that the enzyme recognizes and binds to. And when that enzyme lands there, it's going to be positioned perfectly so that it can transcribe the yellow region of the DNA.